There is a repair crew inside your body that only shows up to work during the night. Not a metaphor, it's documented biology. While you sleep, and specifically during the deep slow-wave phases of sleep, a set of cellular processes runs at its peak regenerative activity. Hematopoietic stem cells renew your blood and immune system. Mesenchymal stem cells repair connective tissue and muscle. Intestinal stem cells rebuild the gut lining that processes your food and houses the majority of your immune tissue. The entire cellular maintenance operation that keeps you functional runs its most intensive shift while you are unconscious. Here is what most people do not know. That crew shows up or doesn't, depending on biochemical signals that are substantially determined by what you eat or don't eat in the 2 to 3 hours before bed. Not in some vague general wellness sense, but through specific documented molecular pathways connecting the timing and content of your evening food intake to the activation of specific stem cell populations. There is a direct line from what you chew before bed to whether your body's repair program runs fully tonight. Listen closely now. The research on how circadian biology, sleep architecture, and nighttime nutrition connect to adult stem cell function is one of the fastest moving areas in cellular aging science. What the last few years of peer-reviewed work have established is considerably more precise than what most people have heard. I am going to give you that precision tonight. The mechanisms, the foods, the specific doses, and the timing the evidence supports. By the end of this video, you will have something you can act on tonight, not next week. As always, this is educational content based on peer-reviewed research. There are specific medication interactions in this video that matter. If you are on antidepressants, blood thinners, or statins, pay close attention to the notes I will flag as we go. Before I tell you what to eat, I need to give you the framework that makes everything else make sense. Because if you do not understand why stem cell function is connected to sleep in the first place, the protocol will seem arbitrary. Once you understand the mechanism, it becomes logical, and things you understand tend to stick. Your adult stem cell populations are the maintenance infrastructure of your body. The hematopoietic stem cells in your bone marrow produce approximately 200 billion new blood and immune cells every single day, a continuous manufacturing operation that never pauses. The mesenchymal stem cells in your connective tissue repair the microdamage that accumulates in muscle, cartilage, and structural tissue through the demands of daily life. The LGR5 positive intestinal stem cells renew your entire gut lining every 3 to 5 days, maintaining the barrier that keeps pathogens out, the absorptive surface that pulls nutrients from food, and the immune tissue that houses approximately 70% of your immune system. All of these populations decline with age. Mesenchymal stem cells fall from roughly 1 in every 10,000 bone marrow cells at age 25 to 1 in every 250,000 by age 65. That is a 25-fold reduction in numbers. And the cells that remain are not just fewer, they are functionally compromised, reduced self-renewal capacity, reduced differentiation range, and an increased tendency toward premature senescence. Hematopoietic stem cells do not decline as sharply in numbers, but they undergo what hematologists call clonal simplification, a narrowing of the diversity of immune cell populations they generate, which directly degrades immune competence over time. Now here is the connection to sleep that most people have never been told, because it comes from research that hasn't filtered into mainstream health conversations yet. The Journal of Experimental Medicine study by Vallet and colleagues in 2022 followed 14 people through control conditions of chronic sleep restriction. What they found surprised even the researchers. Restricted sleep didn't simply reduce stem cell activity, it actually increased the division rate of hematopoietic stem cells by a factor of 1.52 to 1.56. At first glance, more division sounds like a good thing. More renewal, more production. But what kind of division matters enormously? Rapid, unorganized stem cell division outside the correct signaling context of deep sleep does not produce beneficial renewal. It accelerates clonal hematopoiesis, the dominance of one or a few stem cell clones at the expense of immune diversity. That state is a documented precursor to hematological disease and cardiovascular risk in older adults. The takeaway here is critical. It is not just how much you sleep, it is the biochemical quality of the environment during deep sleep. That environment determines whether the stem cells activating overnight are doing organized beneficial regeneration or uncoordinated, potentially harmful overdivision. And that biochemical environment is substantially shaped by what you consumed in the 2 to 3 hours before you closed your eyes. The central mechanism connecting what you eat before bed to stem cell function involves melatonin. I want to explain this with the precision it deserves, Because most people know melatonin only as the sleep hormone. What it does inside stem cells is a different story entirely. Melatonin is produced primarily by the pineal gland in response to darkness. Its production begins 1 to 2 hours before the environment fully darkens, peaks in the early morning hours, and disappears with light. Most people know that profile. What most people do not know is that melatonin has receptors on cells throughout the body, including direct receptors on mesenchymal stem cells, and when melatonin binds to those receptors, something specific happens at the molecular level. The study by Ho and colleagues, published in the International Journal of Molecular Medicine in 2019, worked with human mesenchymal stem cells derived from adipose tissue, the most clinically accessible MSC source, and documented two findings that matter enormously for anyone over 50. First, melatonin binding to MSC receptors promoted cell proliferation by inducing the expression of SOX2. SOX2 is what cell biologists call a stemness transcription factor. It is the gene that keeps stem cells being stem cells. It maintains their capacity for self-renewal and for differentiating into multiple cell types when tissue repair requires it. When SOX2 expression is low, stem cells mature prematurely into a single specialized type and lose the regenerative versatility that makes them valuable. Melatonin keeps SOX2 expressed. It keeps the stem cells as stem cells. Second, melatonin inhibited replicative senescence in MSCs through the activation of SIRAT1. You may have heard of SIRAT1 in the context of NAD+ and aging. It is one of the primary anti-aging SIRAT1 proteins. When melatonin activates SIRAT1 in mesenchymal stem cells, it delays their entry into cellular senescence. Why does that matter? Because senescent cells do not just stop working. They secrete what scientists call CSP, the senescence-associated secretory phenotype, a toxic mixture of inflammatory cytokines and proteases that damages neighboring cells and drives nearby healthy cells into senescence themselves. Every MSC that enters senescence prematurely doesn't just stop contributing to repair, it starts actively polluting the cellular environment around it. Melatonin slows that process. The systematic review published in PMC in 2025 consolidated the broader picture across multiple independent research groups. Circadian rhythms and melatonin regulate the proliferation and differentiation of stem cells in skin, intestine, the hematopoietic system, and the brain. This is not one isolated study. It is a converging body of evidence from multiple directions pointing to the same core mechanism. Now here is the problem that makes all of this especially urgent for your age group. Endogenous melatonin production falls dramatically after 50. A person of 65 produces between 60 and 75% less melatonin than they did at age 25. The primary cause is the progressive calcification of the pineal gland, a process that begins in adolescence and advances continuously with age. By the mid-60s, many people's pineal glands are secreting a fraction of what they once did. The repair crew's activation signal, melatonin, arrives at the stem cells each night as a whisper compared to the clear signal those cells received decades ago. The stem cells are still there. The receptors are still there. The machinery for SOX2 activation and SIRAT1 engagement is still there, but the signal triggering all of it has been reduced by up to 3/4. The construction crew is at the site, the phone just isn't ringing. This is why nighttime nutrition matters. Biologically, not as a vague wellness concept, but as a direct intervention. There is a dietary compensation pathway for the declining pineal gland. It operates through the amino acid tryptophan, and it only works if you use it at the right time. The conversion pathway works like this. Tryptophan absorbed from food enters the bloodstream, crosses the blood-brain barrier, and arrives at the pineal gland. During daylight hours, with light-driven serotonin production dominating, tryptophan converts preferentially into serotonin. When darkness falls, the enzyme aryl-alkylamine N-acetyltransferase activates in the pineal gland and redirects the pathway. Serotonin converts to N-acetylserotonin, which HIOMT then converts to melatonin. Magnesium is a required cofactor for that final enzymatic step. The timing implication is everything. Tryptophan consumed 2 to 3 hours before bed arrives at the pineal gland, precisely when darkness is going to activate the conversion pathway. The same tryptophan consumed at noon goes predominantly towards serotonin, not melatonin. It is not a small difference in efficiency. It is the difference between delivering raw material when the factory is running a night shift versus delivering it in the middle of the day when that line isn't even open. Same nutrient, same food, completely different biological outcome depending solely on when you consume it. Let me walk through the specific foods with the precision this deserves. Pumpkin seeds are the highest yield tryptophan source in the whole food category by a significant margin. At approximately 576 milligrams of tryptophan per 100 grams, a 28-gram handful, about what fits in your palm, delivers roughly 160 milligrams of tryptophan in the melatonin synthesis window. Beyond tryptophan, pumpkin seeds contain magnesium and zinc, both of which are direct cofactors in the melatonin synthesis pathway. When you eat a handful of pumpkin seeds before bed, you are not just delivering the substrate, you are delivering the substrate, the enzymatic cofactor, and the mineral. The final conversion step requires simultaneously. The practical format is plain, lightly toasted if you prefer. A small bowl 90 minutes before bed, nothing elaborate, no preparation required. Walnuts provide approximately 150 milligrams of tryptophan per 30-gram serving, alongside alpha-linolenic acid, the plant-based omega-3, which contributes to the phospholipid composition of cell membranes in stem cells. Healthy cell membranes improve receptor function, including the melatonin receptors on MSCs that this whole mechanism depends on. A 30-gram portion, eaten plain or mixed with a small portion of plain yogurt, provides the tryptophan contribution without the digestive burden that would disrupt sleep quality. Ripe banana deserves special mention because of what accompanies the tryptophan: vitamin B6. B6, specifically pyridoxal phosphate, is a direct cofactor in the enzymatic conversion of tryptophan to serotonin, and from serotonin toward N-acetylserotonin. The riper the banana, the higher the B6 bioavailability. A medium banana consumed 60 to 90 minutes before bed provides both the tryptophan substrate and a key cofactor the pineal gland needs for efficient conversion. For people who find larger evening food intake disrupts sleep, this is the lightest touch option on the list. Low volume, high biological utility. A small portion of turkey or chicken breast, 60 to 80 grams, eaten as part of dinner, 2 to 3 hours before bed, contributes meaningfully to the evening tryptophan pool. Turkey has among the highest tryptophan concentrations per gram of any common food. The cultural association between Thanksgiving turkey and sleepiness reflects a real mechanism, even if the dose from a single meal is modest. The key here is the timing. As part of a dinner eaten 2 to 3 hours before bed, not as a late snack immediately before lying down. The protein load needs time to process before sleep. Tart cherries are in a category by themselves because they operate through a mechanism that the other foods don't. The study by Burkhardt and colleagues, published in the Journal of Agricultural and Food Chemistry, documented that tart cherries, specifically the Montmorency variety, do not just provide tryptophan as a melatonin precursor, they contain directly bioavailable melatonin. 30 milliliters of concentrated tart cherry juice delivers approximately 13 to 17 nanograms of bioavailable melatonin. That is not a pharmacological dose. Pharmaceutical melatonin supplements are measured in micrograms to milligrams, but it represents a meaningful dietary contribution to circulating melatonin levels during the critical sleep window. The anthocyanins in tart cherries, at a third layer, controlled clinical trials using objective polysomnography measurements, rather than self-reports, have shown that tart cherry consumption objectively increases time in deep slow-wave sleep, not total sleep duration, but specifically the deep phase. That distinction matters here, because deep sleep is exactly when the stem cell regenerative activity this protocol is designed to support reaches its maximum. The tart cherry option gives you tryptophan as a precursor, directly bioavailable melatonin, and polyphenols that improve the architectural quality of the sleep phase where the repair work actually happens. It is the most mechanistically layered choice on this list. 2 interactions to flag clearly. Tart cherry juice has significant natural sugar content. People with elevated blood glucose or diabetes should dilute the juice substantially and monitor their glucose response. Second, tart cherries modulate the CYP3A4 enzyme system, which affects statin metabolism. If you take a statin, discuss this interaction with your physician before adding tart cherry juice. It is manageable, it is not a prohibition, but it requires an informed conversation. The overnight fast is the second mechanism in this protocol, and it operates on a completely different stem cell population, through a completely different pathway. Where the melatonin mechanism acts on mesenchymal stem cells, the overnight fast acts on hematopoietic stem and progenitor cells, HSPCs, by removing the insulin and IGF-1 signaling that keeps them suppressed. The most direct human evidence on this comes from the research by Abdel Hadi and colleagues at the Abu Dhabi Stem Cell Center, published in Stem Cells and Development in 2025. 10 volunteers observed Ramadan fasting, 30 days of daily fasting with blood samples collected twice daily on days 0, 10, 20, and 30. The finding was unambiguous. Fasting measurably increased the absolute count of circulating HSPCs carrying the markers Lin negative and CD34 or CD133 positive. These surface markers identify the most primitive hematopoietic stem cells, the ones with the greatest self-renewal capacity and the broadest regenerative potential. Fasting mobilized them from their dormant niche in the bone marrow into active circulation. Not a pharmaceutical, not an injection, the absence of food. The mechanism explains why hematopoietic stem cells at rest in the bone marrow are held in their dormant state partly by insulin and IGF-1 signaling. When blood glucose and insulin are chronically elevated, as is increasingly common with the insulin resistance that develops with age, those inhibitory signals are continuously active. The HSPCs are receiving a constant biochemical message. Resources are abundant, conditions are stable, stay conserved and dormant. A 12-hour overnight fast lowers insulin substantially, reduces IGF-1 toward its morning nadir, and removes that inhibitory pressure. The HSPCs receive a different signal. Resources are constrained. Activate the self-renewal program. The Brandhorst study from Walter Longo's laboratory at USC, published in Cell Stem Cell in 2014, established the broader principle underlying this mechanism. Fasting cycles activate a regenerative switch in hematopoietic stem cells through IGF-1 and pKa suppression. The paper documented immune system regeneration in mouse models and protective effects in cancer patients undergoing chemotherapy. The 12-hour overnight fast does not replicate the dramatic effects of a full 48-hour extended fast. That is a different protocol for a different video. But the overnight fast establishes on a nightly basis the low insulin, low IGF-1 environment that allows HSPCs to function with less chronic inhibition. Every night consistently, you are removing the chemical brake that aging and insulin resistance have been applying to your hematopoietic stem cells. Practically, this translates to one clear rule: finish eating before 8 p.m., ideally between 7 o'clock and 8 o'clock, and do not consume anything more until breakfast. Plain water and herbal tea without sweeteners do not interrupt the metabolic fasting signal. The nightly glass of wine, the piece of chocolate, the glass of juice, each of these breaks the fasting window and re-elevates insulin, reinstating the inhibitory signal on HSPCs within minutes. This requires consistency more than rigidity. 3 to 4 nights per week of maintaining the window produces meaningful cumulative benefit. 5 to 7 is the goal. The composition of dinner matters too, and it connects directly to this mechanism. Protein-rich meals stimulate glucagon, which has metabolic effects broadly opposing those of insulin, and does not suppress HSPCs in the same way. Refined carbohydrates at dinner, large portions of bread, white rice, pasta, or any dessert, generate the insulin spike that directly undermines the overnight fast's ability to reduce HSPC inhibition. Protein and vegetables for dinner, keep the carbohydrates you need low glycemic. The third mechanism operates on intestinal stem cells, and it involves a concept many people have heard of, but few have connected to stem cell biology. Circadian timing. Intestinal stem cells are the most actively dividing stem cells in the adult human body. The entire small intestinal mucosa, every cell of the lining that absorbs nutrients from your food, maintains the barrier against pathogens, and interfaces with 70% of your immune tissue, is completely replaced every 3 to 5 days. That is an extraordinary renewal rate, and it is entirely dependent on the LGR5 positive intestinal stem cells in the crypts of the intestinal wall keeping pace. Research reviewed in the 2025 PMC Systematic Review established something precise about how these cells work. Their cell cycle is directly regulated by the circadian clock genes, CLOCK and BMAL1, which control the G1 to S phase transition, the checkpoint at which a cell commits to replicating its DNA in preparation for division. This means intestinal stem cell renewal is not random, it is scheduled. It is organized around a biological rhythm that your behavior either reinforces or disrupts. When meals are irregular, when eating happens late at night, or when bright artificial light prevents the melatonin signal that anchors the circadian rhythm, the clock and BMAL1 regulation of the ISC cell cycle loses coherence. The intestinal stem cells still divide, but on a disrupted and uncoordinated schedule. The long-term functional consequences are less efficient mucosal renewal, reduced barrier integrity, more intestinal permeability, and more variable nutrient absorption. Most people over 60 who experience digestive inconsistency, energy fluctuations after meals, or foods that used to agree with them and now don't, are experiencing the downstream functional effects of ISC circadian disruption. They attribute it to age. The mechanism is more specific than that. The most effective circadian signal for intestinal stem cells is the consistent timing of the last meal of the day. Eating dinner at the same time each evening during declining light conditions gives the clock BML1 system the environmental cue it needs to stay synchronized. Not rigid to the minute, but within a 30 to 60 minute window that is consistent enough for the biological rhythm to organize around. It is a simple zero-cost intervention that directly supports the stem cells managing your gut health every day. Here is the complete evening protocol assembled from these three mechanisms. Between 7 o'clock and 9 p.m., as part of dinner or immediately following it, consume one tryptophan-rich option from this list. You do not need all of them. Pick the one that fits most naturally into your evening. A 28-gram handful of pumpkin seeds delivers tryptophan, magnesium, and zinc simultaneously. This is the most complete single food option for melatonin synthesis support. A 30-gram portion of walnuts provides tryptophan alongside omega-3 fatty acids that support cell membrane quality in stem cells. A medium-ripe banana provides tryptophan and B6 in a low-volume format that does not burden digestion. 60 to 80 grams of turkey or chicken breast, as part of a protein-forward dinner, contributes to the tryptophan pool without requiring any additional evening food. Or, and this is the option with the most layered mechanism, 30 milliliters of concentrated Montmorency tart cherry juice diluted in 150 milliliters of water. This provides a tryptophan precursor, directly bioavailable melatonin, and deep sleep-enhancing anthocyanins in one small glass. After that last food intake, the rule is simple: nothing until breakfast. Water and unsweetened herbal tea do not interrupt the fasting signal. Everything else does. The target is 12 hours minimum between the last food and the first food of the next morning. 30 minutes before bed, take magnesium glycinate at 200 to 400 milligrams. Magnesium serves two distinct functions in this protocol. It is a required cofactor for HIOMT, the enzyme that performs the final conversion from N-acetylserotonin to melatonin in the pineal gland. Without adequate magnesium, the conversion is enzymatically limited at that final step, regardless of how much tryptophan you have provided. Separately, magnesium glycinate objectively improves deep slow-wave sleep, documented across multiple sleep quality trials. Not total sleep duration, but specifically the deep phase where maximum stem cell regenerative activity occurs. The glycinate chelate form has the best gastrointestinal tolerability, especially relevant if you found other forms of magnesium too laxative. One note on exogenous melatonin supplementation, because many people in this audience use it. The dosing evidence is counterintuitive. Most supplements are sold in 3 to 10 milligram doses. The receptor pharmacology research suggests that 0.5 to 1 milligram is both sufficient and substantially safer for chronic use. Higher doses can desensitize MT1 and MT2 receptors over time, producing tolerance and ultimately reducing your sensitivity to both supplemental and endogenously produced melatonin. If you use exogenous melatonin, 0.5 milligrams is the evidence-matched dose. Do not combine it with benzodiazepines. Warfarin users, the anticoagulant interaction is documented. Discuss with your physician before starting. And if you take antidepressants in the SSRI or MAOI class, discuss any change in tryptophan intake with your prescribing physician before implementing this protocol. The serotonin pathway involvement makes that conversation necessary. Let me address the questions I know this raises. If you are naturally hungry in the evening and feel like the tryptophan food is too small, the protocol isn't asking you to be hungry. It is asking you to eat dinner adequately earlier and then have a small tryptophan-rich option afterward. A substantial dinner at 6:30 or 7 p.m., followed by a handful of pumpkin seeds at 8:30, satisfies both hunger and the mechanism. If you have been eating late for decades and wonder if there is any point in changing now, the ISC circadian research shows that the biological clock begins resynchronizing within 1 to 2 weeks of consistent timing changes. The HSPC mobilization data from the Abu Dhabi study showed measurable increases in circulating stem cells within 10 days of consistent fasting. These systems have not stopped responding. You are sending new signals to machinery that still responds to them. If you are wondering whether the occasional late meal, the holiday dinner, or the social exception breaks everything, It doesn't. Consistency matters more than perfection. 3 to 4 nights per week of the full protocol produces a meaningful cumulative effect. The exceptions are exceptions in a pattern. They do not erase the pattern. What should you expect and when? Days 1 to 3. Most people notice improved sleep quality first. Specifically how they feel upon waking. Not necessarily longer sleep, but qualitatively different sleep. More time in the restorative deep phase. Less fragmented. This is the first sign that the melatonin biochemical environment during sleep is improving. Week 1 to 2. Morning stiffness typically begins reducing. This is one of the most sensitive functional indicators of systemic overnight inflammation, and it corresponds directly to reduced SAP output from MSCs that are receiving better melatonin signaling and entering senescence less rapidly. Days 10 to 14. The HSPC mobilization research in the Abdel Hadi data shows measurable changes in stem cell circulation in this window, with consistent nightly fasting. Functionally, this often manifests as more stable and consistent morning energy. The intestinal stem cells are renewing the gut lining more efficiently, which improves nutrient absorption from the first meal of the day. Month 1. If you have baseline blood work, inflammatory markers, particularly high-sensitivity CRP, typically begin trending downward. This corresponds to reduced niche toxicity from MSCs receiving improved melatonin protection. Months 2 and 3. The cumulative tissue regeneration effects become apparent in the most functional measures: wound healing speed, immune response quality, and recovery from physical stress. These changes are slower because structural tissue changes take longer than blood chemistry changes, but they are real and they are the ones that matter most for sustained quality of life.